Okay, so now, first of all, we want to study the role of sensor and transducer in biomedical instruments. Of course, in the first hour, I have told you some of the, in a glance, yeah, what are the role of sensor and transducer in biomedical instrumentation. Basically, the sensor and transducer will be the forefront, yeah, of any instrumentation. It capture the actual physical, or could be also chemical, could also be biological informations into electronic information, into electronic signals. So these are the current role of sensor and transducer. We don't know in the future, maybe electronics will not be the main information carrier. Maybe people will switch to photonic, for example, the use of light. Then maybe the sensor and transducer will also be different. But at least for now, we study electronics and we study the sensor and transducer that converts all these different forms of information into electronic form. Okay, now, okay. Apa jadinya ya? Pen. Okay. Okay. Oops. Sorry. Wait a moment. Share. So the sensor and transducer in biomedical instrument, as I mentioned, it is located in the front, yeah, of any instrumentation system. It captures the physiological and pathological states of the human body, of the subject's body here, and in the form of, we call it biosignal, in the form of biosignal, and converts it, this part, the sensor and transducer, converts it into raw electrical signal. The raw electrical signal later can be further processed, either using analog or digital signal processing. Yeah, in the analog means when we still use voltage, current, and other forms, yeah, using amplifier, resistor, capacitor, etc. But when it is already converted using analog digital converter into digital signal 010101, then we can process it further using microprocessor, for example, or microcontroller. And the digital process signal can be later displayed or analyzed, yeah, displayed in a display like this, or further analyzed, recorded. We can also process digitally also, yeah, using filtering also, or amplification digitally, and many other kinds of signal processing in digital form. So here, the role of sensor and transducer, again, what we are studying in this subject, in this course, is the main device in the forefront of any biomedical instrumentation system that can convert biosignal into raw electrical signal. Of course, in later development, in the recent study, even the processing here can be integrated with the sensor to form what we call as intelligent sensing system. Okay. Okay, so for example, this is an example of ECG recording. Yeah, ECG recording. Here, can you mention then where is the sensor? Anyone?
The one stuck on the chest.
Yes. Tanita ya tadi ya?
Yeah.
Yes. This will be the sensor, yeah. So this can be called or can be mentioned as the sensor, yeah, because it transduces the signal from the body. In this case, it is the bioelectricity. In the bioelectricity of the body into electrical signal, raw electrical signal, and then convey it to this analog processing system. Yeah, in this case, maybe there is no, there is not a lot of digital processing. Instead, it immediately records or in the form of charge. Yeah, so there is no display, for example. Okay. This is another example of active belt. This is the kind of biomedical instrument that can measure. It has this textile electrode. It can measure also the ECG signal, but in the wearable ECG. Yeah, and then it also includes integrated into the belt the amplifier as well as analog digital converter, which will then send the data, the digital signal data, to dual core embedded system, dual core processor embedded within this belt. And then from here, send the signal wirelessly to another workstation, either computer or either computer or smartphone, for example. Okay, so this is how it looks like. This is one form of this active belt, but implemented for babies. Neonatal smart jacket. It has this textile electrode here. The black part is the textile electrode. It also measures the EEG of the baby, yeah, in the form of this cap electrodes. Yeah. So from here the second part here is the ECG, electrocardiography from the heart. The upper part here, what is it? Yeah, I think ECG as well, but from different electrodes. Yeah. This is the left arm ECG. This is the right arm ECG. And then we have this ACM X and Y and Z. I think this is EEG, the electroencephalography from the brain of the infant. Okay, okay, reset pointer. Yeah, then this is, if you want to further information, you may have a look at these references. Yeah, Bostra. Maybe I will put it if I have it. I think I have it. I will put also in the Google Classroom, yeah, so that you can have read on these references. Okay, next. So these are some basic concepts. Yeah. So the sensor or transducer here is a device which can respond to a measured object, yeah, and transform it into signal which can be detected. So in this case, most of the time, it will be electronic signal. Then the electronic signal can be processed further. Again, respond to a measured object and transform. So there is a transformation signal here. Okay, there is a transformation signal. Okay, yeah, transform means there is a transformation, or you can also call it conversion of the signal form. For example, in the ECG case, it will be from bioelectricity into electronic. Okay, a sensor is usually composed of a sensitive component, which directly correspond to a measured object. Here, sensitive component that responds to the measured object, a conversion component. This is what we call as usually the transducer part. Yeah, as the name implies, transducer means to transduce. Trans means to go across, right? So transduction, to go across from one form of energy to another form of energy. Information is basically energy. Okay, information is basically also energy. But what is the difference then between information and energy? In the information, we are more concerned about the quality of the energy rather than the quantity. So quality instead of quantity. What is the meaning here? For example, in the case of lamp, yeah, you know, lamp lighting equipment, um, it converts electrical signal into radiation. into lighting, right? Radiation energy. What we care is how much light can be produced by the lamp, even though with as small as possible power of the electricity, right? So that is what we concern is quantity in the case of lamp. That is why lamp is not a sensor or an actuator, a transducer in form of information actuator. No, because it is basically an energy converter. Similarly, like with solar cell. With solar cell, it converts light signal from the sun into electricity, right? We don't really care about how the sun looks like. Yeah, but we care is how can we convert as much as little energy from the sun into as much as power as possible of electricity, right? And it means solar cell in that case, in that particular case, can be called as an energy converter. But look now, for example, with the case of camera, right now I'm using camera, so to convey my image, the image of my face to you. So with camera, we are not really concerned about the brightness, as long as it is bright enough, right? You don't really need to really have the camera to have my face too bright, right? If it is too bright, then also you will lose the information. What will be the information? The information here is the shape of my face, the texture of my face, that there is nose, there is mouth, there is eye, there is this eyelids, there is glasses, etc. Those are information. The states of the energy, how the energy is being arranged. Now, this is the quality of the energy, which forms the information. With camera, what we care is how can the states of the energy in the form of this face can be captured by the image sensor in the camera, converted into electricity, into electric signal, then conveyed through the computer to the internet, goes to your laptop. To your laptop or camera or handphone, there is this monitor. Yeah, the monitor also, you don't really care about how bright the monitor is, right? What you care is as long as it is bright enough that it can display my face, that information of my face in your camera, as it is clearly with the texture clearly, then it is enough. So that is quality of energy, which is named as information. So quantity here, if you review back your physics lesson from Pak Fatoni, quantity here is dealt with the first law of thermodynamics. Yeah. While quality here is in the second law of thermodynamics. Second law of thermodynamics deals with entropy, yeah, the states of energy. That is what we are dealing with. Information. Information is the states of energy, yeah, from philosophical point of view. So sensors often provide information about the physical, chemical, or biological state of a system. Yeah, in this case, the energy structure. Yeah. So it's not the quantity of the energy, the physical, chemical, but the state, the configuration of that energy, and the entropy of that energy is what is being captured by the sensor. Measurement here then is defined as an operation that aims to obtain the measured value of a quantity. So for example, in my case, in this camera case, again, what is the texture of this face is represented by the intensity of the brightness intensity of different points, pixel within the camera, yeah, represented as red, green, and blue, the basic color, right? When it is then being again transduced into light in your screen, it can represent my face back. Okay, that value, the RGB value in the case of camera and monitor, but in the case of thermistor, it will be temperature value. Yeah, how many degrees of Celsius, how many degrees of Kelvin, etc. Sensors are defined as devices that can transform non-electrical signal into electrical signal. So in this case, we limit. This is of course actually already very limited definition. In the brighter sense, actually sensor can also mean any signal form into another any signal form. Okay, but since nowadays in this current modern world, we mostly process information in the electrical domain. That is why the definition becomes like this: non-electrical signal into electrical signal. We don't know in the future. In the future, as I mentioned earlier, it could be in the light form, what we call as photonics. Then maybe non-light form, non-light signal, non-photonic signal into photonic signal. Then biomedical sensors are special electronic devices which can transfer various non-electrical quantities in biomedical fields into easily detectable electrical quantities. So non-electrical quantities in biomedical fields. Yeah, for example, pressure, ionic. Yeah, bioelectricity is basically ionic measurement. The transfer of ions within our body. It is bioelectricity. Biomagnetic into detectable electrical quantities. Okay, so far understood, yeah? I hope. Okay, now classification of biomedical sensors. So according to the working principle, we can classify biomedical sensors into physical sensors that work based on the physical nature and effect. Yeah, for example, as we have discussed or we have mentioned earlier in the first hour, resistive sensor, yeah, resistance strength sensor, semiconductor piezo resistive sensor. Piezo means that physical law that relates physical strain or pressure with resistance change. Yeah, this is piezo resistivity. Piezoelectric sensor, a law that relates piezo or pressure change, stress change into voltage signal, yeah, into electricity. Photoelectric sensor, when there is radiation change into electricity. It can do, biomedical sensor can also be classified as chemical sensor that work based on chemical effects. Yeah, like ion selective electrodes, ion sensitive tubes, humidity sensors, and etc. So this converts chemical change, yeah, in the field, in the body, into electronic change, yeah, by the sensor. So biological sensors or biosensors work by using, so I should actually underline this as well. Yeah, biosensor, this work by using biological active materials as a molecule recognition system. As also I have mentioned in the first hour, yeah, enzyme sensor that use enzyme, microorganism sensor using bacteria, for example, can be as a biosensor. Yes, there is sensor that use bacteria, for example. Immunity sensor, this uses antibody. Tissue sensors use, for example, a tissue from animal, yeah, as sensing material. DNA sensor, this is using DNA probe. Yeah, the DNA sensor using genetic material as the biomolecule to sense any biological measurement, biomedical. So we can classify biomedical sensors based on these three, based on what phenomena that they are capturing. Is it of physical nature, like pressure, temperature, what else? Stress, strain, acceleration, yeah, or radiation. These are all physical phenomena. Then we can call them as physical biomedical sensor. If it were chemical composition, chemical compound, chemical energy, then they are chemical sensors. If what we want to measure is biological compound, biological structure within our body, then biosensors or biological sensor. Okay. Based on the detection type, yeah, based on the detection type, it could be displacement sensor. Yeah, displacement sensor measure the displacement. Flow sensor measure the flow of certain material within the body, blood flow, gas flow. Temperature sensor, yeah? Like thermal resistance sensor, thermocouple sensors, p-n junction temperature sensor, and other sensors that can detect temperature. Speed sensor, measuring the speed. Pressure sensor, yeah, like metal strain foil pressure sensor, semiconductor pressure sensor, capacity pressure sensor, and other sensors that can detect pressure. Okay. Then for the biomedical measurement technology, this will involve the detection of physical, chemical, or biological signal, as we have mentioned in the previous slide, in different levels of organism. It could be physiological level. Physiological level means the whole body, the whole body physiology. Yeah, in the case of electrical physiological signal, the example will be ECG. Yeah, this is from the heart or originates from the heart, but actually we can measure it. We measure it by putting the electrodes in the end of our left arm finger, right arm finger, also left leg, right leg, some electrodes in the chest, some electrodes in the stomach, etc. Electroencephalography, yeah, especially in the brain, in the head. Electromyography in the muscles, in the particular muscles we want to measure its activity in the hand muscles, leg muscle, etc. Non-electrical physiological signals. So these are physiological signal from the body, but the nature is not ionic, but rather other physical phenomena like blood pressure, pressure of the blood, the temperature of the body, the breath, yeah, the flow of the breath, the volume of the breath, blood flow, and the pulse coming from the heartbeat. Then chemical signals, yeah, the chemical content of the blood and urine, for example. But we can extend this to other body fluid, chemical content of saliva, chemical content of sweat, chemical content of tears. What else? Other body fluid, chemical contents of feces, yeah, chemical content of tears, mucus, for example, yeah, mucus, and many kinds, different types of body fluids from our body, which basically they reflect the physiological condition of the subject. Yeah, so this contain chemical signals. Then biological signals relates to the biological materials, biological compounds contained within our body. Yeah, for example, inside our blood, what enzymes are there? What proteins are there? Do we have the antibody for SARS-CoV-2 virus, for example? It is a biological signal. Are there the SARS-CoV-2 virus in our saliva? It is antigen signal, for example. Yeah, these are biological signals which necessitates the use of biosensor in order that we can detect them. Okay. So from these examples that we have mentioned of biomedical sensors and measurement, we can say that this field is an interdisciplinary field of research because even one sensor, one biomedical sensor, to develop them requires the knowledge of the physiological signal itself, some knowledge about physics, yeah, some knowledge maybe of chemistry and biology if it is chemical sensor or biological sensor, and some, of course, knowledge on electronics because the signals is converted into electronical signal, after which we need to process them. Then if we want to develop a system that also interprets the signal, then some knowledge on information technology or artificial intelligence is also required. Then it's also relates with basic research and technological innovation. Yeah, with the development of new techniques, new science, with nano technology, for example, now more and more sensors are being made smaller and smaller, yeah, to include nanotechnological development into the sensor itself. So previously, the sensor very big size, you need a whole room, for example, to develop such sensor. Then they become smaller. As I mentioned in the previous hour, genomic sequencing, previously you need one big table to put the equipment, but in nowadays it could be made into one small pen like this, yeah, because it already involve nanotechnology to develop the measurement system. And sensitive materials and film formation technology. Sensitive materials here, the materials that can be sensitive to particular physical signal. So for example, in the case of thermoelectricity sensor, we need thermoelectric material that will change its potentials upon receiving temperature difference, for example. Film formation here means that the material is being coated as thin film, as thin layer of material above certain substrate. So they are not in the bulk form anymore. So why film formation? Because then the sensitivity can be increased, plus the size can be made miniaturized. And this field of biomedical sensor measurement is very knowledge intensive. As I mentioned, it requires a lot of knowledge in both the application as well as in the development of the material, and then later also in the processing of the signals. Yeah, high reliability. Yeah, because we are working in biomedical. The application is biomedical related to human. You need to have a reliable sensor in order that you acquire reliable information. Because without reliable information, then you cannot provide reliable information for the physician, with which the physician will determine the appropriate, adequate treatment, yeah, for that subject or patient. Fine technology means very precise technology now, yeah, not random technology. So special requirements for biomedical measurements, among others, like it has to be non-invasive, safe, and reliable measurement. Yeah. Involve some special measurement methods, low noise and anti-interference technology, because why it needs this method? Inside the body, there are many sources of noise. Even our own movement can be a source of noise for the sensor. Even the surrounding electronic equipment can be a source of interference noise for our sensor. Yeah, picking up signals, analyzing and processing technology and measurement system, measurement using analog and digital circuitry, computer hardware and software, and sometimes brain-computer technology, brain-computer interface technology. Then when the sensor is attached to our body or implanted inside our body, then we need to consider also biocompatibility issue. This is you will learn from biomaterial subject, yeah, from Pak Holis later, hopefully. Yeah, among others, hemocompatibility, compatibility with the blood, histocompatibility, compatibility with the surrounding tissue. Then manufacturing requirements, yeah, sometimes with more complex biomedical sensors, the manufacturing requirement will also be more complex. What are the example of this manufacturing requirement? For example, the material selection. If you use metallic materials in sensors, it has to be inert metal because it will be put inside the body, for example, like stainless steel or titanium alloys. If you choose polymers, it has to be degradable, yeah, such as PMMA or silicones. A series of experiments on animals and clinical trials should be carried out before clinical applications. This is what applies for example, GNOS. Although GNOS does not include any more animal trials, it directly is being tried with humans subjects, yeah, in various hospitals. This is when and they are doing the testing of genomes, yeah, before they do application with real patients of COVID-19. Application of biological methods to evaluate the host response. The in vivo biocompatibility can be evaluated by analyzing the cell population present there, measuring the mediator and metabolite cells excreted, and analyzing the morphologic characteristic of the tissue and the capsule thickness around the implant. So this is if we are developing implantable biomedical sensor. Yeah, we check the biocompatibility using these three different category. Yeah, the cell population near the implant itself. What is it? How is the condition? What are the mediator and metabolite cells being excreted? Yeah, after we introduce the implant, are they signs of toxicity or not? Then the characteristic morphological characteristics of the tissue. Usually we obtain this using microscopic histological study, whether they form kind of immune response, yeah, due to that strange form of material, the biomedical implantable biomedical sensor introduced into that tissue. An appropriate immobilization on the sensor surface is also required for maintaining biological viability and activity in biosensor. So if, as we have introduced in the previous slide, that in biosensor, we use biological molecules. Yeah, it could be enzyme, could be antibody or antigen, could also be DNA, RNA, aptamer, or cells, the whole cells, bacteria, etc. And we usually put them into a substrate material which will act as the physical transducer. This biological thing, biomolecule or whatever, will transduce biological signal into another physical signal, let's say weight change, mass change, and then this physical signal will be transduced by the corresponding physical transducer onto which the biomolecule is attached into electronic signal. Okay, and the way we put this biological molecule or biological things, yeah, onto the physical transducer is called as immobilization on the sensor surface. And this is important, how we immobilize, so that then we can capture the correct biological signal. Okay, so this is an important step in the manufacturing of biosensor. So this is the trend, yeah, in biomedical sensor and measurement. Previously, the trend is from invasive testing towards non-invasive testing. Yeah, now, for example, in glucose blood sensing, previously we have to puncture our finger, take some small blood sample, drop it onto this paper-based test kit, and then the cassette is put into the instrument, the small instrument. This we can purchase, yeah, in Dr. Watson or Guardian if you want, or in any apotheke, pharmacist. Then you get your blood glucose level. But there is a tendency, for example, to do non-invasive because people do not like their fingers to be pricked all the time. Yeah, maybe if you are healthy, you don't prick too often. But if, God forbid, if you have diabetes mellitus, sometimes you need to do this 3 times. Hopefully, all of us are healthy. No one has diabetes mellitus. But I knew when I have one student whose mother has, I used to have one student whose mother has diabetes mellitus. And she needs to do this 3 times a day, so that then she can monitor the level of the blood glucose. Yeah. So when it is necessary, she will need to inject herself the insulin. Yeah. So how to do this non-invasively? For example, people are developing non-invasive thing. So for example, why not we use other biofluid to measure the blood glucose level? Instead of pricking blood from our finger, why cannot we use, for example, saliva, yeah, or tears, maybe, or sweat, or urine, yeah, Or here, for example, can we measure the blood glucose level from the absorbent or reflective radiation level at particular wavelength. So basically, this is a pulse oximeter. Yeah? But similar principle can be used for blood glucose level using the absorbed radiation at particular wavelength. Because if the glucose has certain spectral absorbance characteristic, yeah. So it absorbs certain wavelength at particular wavelength specifically. So we can use that property to develop a non-invasive blood glucose sensor. There is also a trend to develop multi-parameters detection sensor. One sensor that can detect different information at one time. So of course, this is not exactly a biomedical sensor. This is a sensor more of environmental sensor. What is the example given in this slide that can measure at one time the pH fluid, then the temperature, and then the other one is this EC. Yeah, what is EC? Yeah, electrical. I forgot. I need to check at one time. But the same thing can be done, for example, when we analyze urine. Yeah, now it is can be conducted using what an instrument called as urine analyzer. You put urine in that sample, then the instrument will give you all the reading from the urine, glucose level, ketone level, protein level, etc. Yeah, this is what we call as multi-parameter detection. In vitro versus in vivo detection. In vitro, you are basically taking the sample first, and then put that sample, process it in the lab, yeah, and then do the analysis in the lab, put in the reaction tube. In vivo is when the sensing is done at that organism. So in this case, can we do, yeah, like I mentioned, the urine, in the current urine analysis, you first have to urinate, yeah, in the toilet. You bring that sample, your urine sample to the lab, and then the lab technician will put that urine into the urine analyzer. Now, can we done it more, instead of we bring the sample to the lab, we bring the lab to the sample to do in vivo analysis. Can we put integrated sensor, for example, into our urinoir or our water closet, so that then we can have more real-time analysis of our urine. Yeah, this is one of the challenge in the near future. Yeah, in silico here refers to the modeling in the computer. This is an example of a product being developed, not yet widely available, what we call as intelligent artificial viscera. This is artificial pancreas. So in the previous case that I mentioned about diabetes mellitus patient, the patient needs to puncture and then analyze the blood glucose level, and afterwards inject himself the corresponding insulin to the blood, to his blood. So in this case, this is an automatized system. There's a sensor here. If I'm not mistaken, this will be the sensor. Yeah, this will be the sensor sensing the transcutaneous glucose level from the blood vessel just underneath the skin. Then it will send the information to this controller of pump. Then based on that information of glucose level in the patient's blood, it will pump the corresponding insulin, yeah, to the blood system of the patient. Yeah, so this is, you may check this website, or you may Google also artificial pancreas. There have been several companies trying to develop this, but they are not yet widely available. And this is the current trend to, as I mentioned also, to miniaturize the sensor using the technology derived from the microelectronics, yeah, to develop micro nano system like this. And now many sensors are being made very small. The collection sample is very small also. Then even the electronics can be integrated within this small chip. Not only the integration happens in the electronic system, but even the fluid processing things can be miniaturized, what we call as now microfluidics, processing the fluid at the micrometer level. Yeah, like this. There are many pumps, reservoir here, reagents are being mixed at some chambers here and then analyzed further. So one single chip can do the analysis that previously have to be done in big instrumentation in the lab. There is also trend towards biomimetic sensor. Sensors that mimic the way the biological things detect or measure their environmental parameters. So because, for example, when sensors are made smaller, if we use the similar principle, usually then the sensitivity will decrease. While some organism like this small fish, they already miniaturized the sensor from far years ago. Yeah, so why don't we copy, not simple copy, but learn the principle from them, try to make similar sensors. So here, for example, the lateral line in a fish that is usually used by the fish to measure the pressure in the surrounding water, as well in that sense, then also the depth, yeah, of the water they are in. So this can be like this. This is how it looks like. Yeah, there's tiny hair cells inside this channel. This can be reproduced using human technology by making this artificial hair structure like this. Yeah, that will bend upon the flow if it is located inside a flow channel. Okay. Exercise indicate. No, yeah, just answer directly. Indicate what is the question like this. The sensor part or the transducer part in the system. Which one is the sensor? This is a sensor. This is a system that can measure the bending of the finger. So here, this is when it is straight. So when it is bent, it changes like this. Yeah? Like up and down, up and down. Anyone? Where is in this picture the sensor part? Please annotate it.
The black lead on the right.
Can you annotate this one? Annotate using your mouse. Siapa? Angeli.
Can I try to answer
Yeah,
it?
siapa Ezra. Okay.
I just think that the sensor is in here.
Can you annotate? Yes. Okay, that's good. Yeah, that is the sensor. Do you know what sensor it is? Can you have any idea?
No, actually.
No idea? Yeah. It is basically made in textile material, and some part of it is being coated with silver nanoparticle. The sensor is made by my former student. Yeah, when undergraduate. Yeah, she is undergraduate student, and she was my student in University Technology Malaysia, UTM. And from here, she published one paper in IEEE Sensors Journal, very good journal. So even you are still undergraduate, you can develop good work, yeah, if you are diligent. And she was a diligent student, one of the best students I've ever had. Okay, then yeah, this is how it looks like. Yeah, this system, also measurement system, is built by herself. Yeah? The time there was not yet any Arduino, but different kind of microprocessor system. So here, when she bent here, then the signal increases. So this is not commercial sensor. This is really built in our lab that time. So, and the signal is transduced into the bending angle of the finger. So the sensor is being sewn into glove, into conventional glove, into I think it is wool, yeah, wool glove. Okay. So again, this is undergraduate work, not works by PhD student or master student. No, undergraduate like you. Yeah, this is using silver nanoparticle being patterned, yeah, along the axis of the finger. Okay. What about this one? Can you pinpoint which one is the sensor and primary transducer? Who else? Angel? Long time no see. Can you pinpoint? Angel Yuswono?
Hello, sir. The sensor is it's in the white thing that she's holding.
Can you annotate using your mouse in the
Uh,
figure? Just like Ezra earlier. So circle the sensor in the primary transducer.
I'm not sure how to annotate, sir. Never mind.
Ada di in the green part here in the
This
screen.
one?
Yes. Okay, good. That's correct. That is the sensor and the primary transducer of this ultrasonography system, yeah, USG system. So in that sensor, there is this piezoelectric transducer that converts. It is basically an active sensor, an active system. First, it transduces electric signal into ultrasonic wave projected into the body, and then the scattering of the ultrasonic wave by the structure of the body is reflected and captured by the same transducer but different part of the transducer. Then reconstructed by the computer as images, yeah, two-dimensional image. But nowadays there is three-dimensional ultrasonography also, and even four-dimensional ultrasonography. Okay, that with time, with showing the three-dimensional structure of the body, elapsed with time. Okay, yeah, that is the last. Indicate the sensor as your homework, processing and display parts of this biomedical instrument. I will put in the slide into the Google Classroom. Just put your answer as one simple sheet, scan, to indicate from this picture which one is the sensor, which one is the processing unit, and the display quote unquote, yeah, so means that it's actually not in the form of visual information but rather in the form of acoustical information. Which one is the biomedical instrument? Of course it is the stethoscope of the doctor. Okay, that will be all for this